The overall goal of the following experiments is to assess the efficiency and specificity of microRNA-based regulation of Picornavirus tropism in vitro. This is achieved by preparing Picornavirus-encoding microRNA response elements in their genome. As a second step, use the viruses to infect cells that express or don't express the cognate microRNAs. Then, evaluate the viral replication kinetics, propagation, and toxicity to determine the efficiency and specificity of targeting.
This targeting method can be used in the field of virology to improve virus safety, manufacturing, and utility, or to facilitate research focused on understanding the biological processes involved in a virus infection. The main advantages of this technique are that it requires minimal engineering of the viral genome, and it's highly versatile because a significant number of cellular microRNA signatures have been identified.
To begin this procedure, plate the permissive cells in a 96-well plate in complete growth media. Add serum-free media without virus to row 1 and row 12 for uninfected controls. Then, incubate the cells at 37 degrees Celsius overnight. Next, make 10-fold serial dilutions of virus each in a total volume of 1 milliliter of serum-free media. Add 100 microliters of serum-free media without virus to row 1 and row 12 on the plate for controls. Tip the 96-well plate to a side and aspirate the media from the plated cells. After that, add 100 microliters of each virus dilution to 8 wells of the 96-well plate. Incubate the plate at 37